Okay, in this video, after watching this, you should understand how stop codons get interpreted at the ribosome. So termination of protein synthesis. We have here the peptidyl tRNA at the P site, and now we're encountering one of the three stop codons, UAA. It could have been U. According to the codon, it could have been UAA or UAG or UGA. Any one of those three codons should be interpreted not with an amino acyl tRNA, but with something that will terminate protein synthesis. So if an amino acyl tRNA comes into one of these stop codons, and this will happen quite frequently. There's just more of these amino acyl tRNAs than there are of the release factors. It's coming in here with elongation factor TU, the clock mechanism, the G protein, and in this case, this anticodon is not going to the base pairing with the stop codon is not going to persist long enough, and this amino acyl tRNA elongation factor TU complex will leave the A site, and it keeps sampling agents that can interpret this until we get a complex with the release factors. And there are two release factors, one whose job is to mimic the interactions with the tRNA, that mimic the interactions that a tRNA molecule would make with the stop codon, and it's in complex with another release factor that is one of these G proteins, and it's in complex with GTP. We still need to include a clock mechanism, because if this is not a stop codon, that clock would give an opportunity for these release factors to leave the A site. But in this case, they will make favorable molecular interactions that last long enough for GTP to be hydrolyzed to GDP and inorganic phosphate. And that then allows this release factor to leave the complex, and that opens the door for water to enter the active site. Normally, water, this H2O molecule, is not included in the active site, and that's in order to protect this ester linkage, which otherwise would be very sensitive to hydrolysis by water. But here, in this situation, assisted by release factor, water does enter and makes nucleophilic attack on this carbonyl center, and what that does is it creates the free carboxy terminus of the polypeptide, which is now able to leave the ribosome. So this becomes the carboxylic acid functional group of the finished polypeptide, and it will leave as a completed unit. And now the ribosome disassembles and goes and looks for a new start codon to start synthesizing a new protein potentially on a different RNA molecule.